A very warm welcome to everyone. Thank you for joining us tonight for our first results seminar for our Exploring Unseen Threats, Contaminant Trends and Impacts in our bays and what our initial monitoring is informing us of. This is funded by the Victorian Government via the Port Phillip Bay Fund. Before we go further, I'd like to acknowledge the traditional custodians of the lands and waters where this project takes place, where we meet today, and to those present. Tonight, Jackie Myers, Palasi Sarasinghe, and myself are leading this presentation, but we'd also like to extend our thanks to our strong volunteer base, community groups, researchers, industry partners, and other stakeholders, all of whom are represented here tonight. We've made a bit of a pie chart to give an idea of the people who have registered to attend tonight. Jack, if you'd like to move the slide on. And just to give you a bit of an idea of who we've got in the room. And shortly, I'll hand over to Jackie. As she takes us through this presentation, if you've got any questions, I ask that you add them to the chat and we'll use them to kick off the general discussion at the end of the talk. So over to you, Jack.
Thanks, Mon. So if people don't know me, I'm Jackie Myers. I'm one of the project leads for this project from RMIT. And I'll be taking you through the webinar tonight. So what we'll be doing is giving you a brief overview of the Unseen Threats program, including how we've engaged volunteers so far, where the program currently stands, and some of the initial results from our first round of sampling. We'll also go over what those results are telling us. We'll share a bit of information on what's coming up next for the program, and then we'll have some time at the end for a bit of a general discussion and questions from anybody. All right, we'll move on. Let's get started. So Victoria's bays and estuaries aren't just beautiful places. They're vital ecosystems that support tourism, fishing, recreation, and community wellbeing. They carry ecological, social, cultural, and economic importance, making them some of our most valuable assets. However, our estuaries and bays are under increasing pressure from urbanisation, population growth, and climate change. And one of the big pressures that they also are under is from contaminants. So thousands of chemicals, right? Like things like plastics, pesticides, industrial compounds are produced every year. And there's always new ones that are being constantly created. And while they make our lives easier, they can harm water quality, marine life, and they can impact on the cultural and economic values of our bays. And so to protect these ecosystems, we need to understand where these contaminants come from, how they move, and the risks that they're posing to those systems. So imagine trying to protect our estuaries and bays without knowing what contaminants are really there or how they might be affecting marine life and the values that we care about. So over the past 40 years, there have been major studies like the Port Phillip Bay Environmental Study and the Westernport Research Program as well that have given us important insights to contaminants occurrence and risks in these systems. But the picture really is still incomplete. And so in the 2021 State of the Bays report, the status of contaminants in estuaries and our bays was rated as fair. And this was basically due to low data availability on these. So we kind of lack some systematic monitoring and we still don't really fully understand the risks to biodiversity, recreation, and cultural values from contaminants. So to manage these risks effectively, we need better information on contaminant sources, their concentrations, and their ecological effects. And so that's where this project kind of fits in. So as Mon said, with support from the Port Phillip Bay Fund, but also some funding from Melbourne Water through our Aquatic Pollution Prevention Partnership, we're working to better understand the occurrence and impacts of contaminants from everyday diffuse sources across our estuaries and bays. Now, collaboration is really heart of this work, and so we're partnering with community volunteers, research institutions, and government agencies who are all contributing to monitoring, protecting these ecosystems. So what we're doing is together, we're monitoring surface waters across multiple sites. We're screening for more than 40,000 contaminants. And sampling takes place several times a year. So we're doing at least 3 times a year over a 3-year period. So this will give us the ability to capture both spatial and seasonal trends in contaminants. In selected sampling rounds, we will also include some toxicological assessments. So this is where we'll take some waters and bring them back to the lab and assess toxicity to flora and fauna. So we're capturing information on the occurrence of contaminants, but also their potential impacts. Now, through the data collection, we'll build a really strong baseline of contaminants of concern in Port Phillip and Westernport estuaries and bays, and this will provide basically an evidence base needed for informed management and also for the long-term protection of these important ecosystems. So how are we doing this monitoring? So as I mentioned, we're assessing surface waters for contaminants, and what we're doing is we're using a device called passive sampler. Now, passive samplers just accumulate chemicals from the water over time, and we're using 2 different types of these. So each one will accumulate different types of contaminants. Now, these samplers are deployed for approximately 1 month out into our estuaries and bay sites. Then they're retrieved, extracted, and analysed by our scientists at the National Measurement Institute in collaboration also with one of our chemists from RMIT. We apply 2 complementary analytical approaches to screen or look at the contaminants that are present. The first is known as a targeted screen, and this one quantifies contaminants from a predefined list that we have of around 450 contaminants, and these are ones that we've sort of built up over time that we've been finding in other sites and locations across Greater Melbourne. Then we also will be doing some non-targeted screening, and this is where we're focusing on completely unknown compounds. And what we do is we match our results to these libraries that contain over 40,000 chemicals. So this is very comprehensive screening, but the non-targeted, it does generate a lot of data and takes a lot more time to process and analyse. So we're doing that on a more select group of samples. Now, I do want to highlight that pollution from contaminants is often an unseen threat. We can't always see it. So unlike something obvious like an oil spill or a surfactant foam, most contaminant pollution is really invisible to us. And a really good example of this is here you can see some of our passive samplers after they were left in the water for 4 weeks. And at first glance, you might kind of look at the sampler on the right, I think it is, the one that looks really dirty. It's heavily biofouled with barnacles, and you might think that might come from a dirtier site than the cleaner looking samplers in the picture on the right. But in reality, when we see this biofouling, that may actually indicate that local species are able to colonize and grow at that site, whereas a clean sampler may reflect a site where some things can't grow and survive. So we might see some of this over time in our samplers, and we'll call this a bit of anecdotal toxicology. And so we'll watch this as we're putting our samples out and retrieving them over the 3 years, and we can make some notes about their condition alongside the chemistry data that we actually pull up and the other toxicological assessments that we'll do. So far, our volunteer engagement has been really incredible. We've had 65 people take part from across the southern coast of Victoria. 30 of you have actually rolled up your sleeves and done hands-on work with us. That's including training sessions, both online and in person, and also getting out there to deploy and retrieve the samplers. Together, our volunteers have already contributed more than 130 hours of effort, which is just amazing achievement. To make sure our volunteers are supported, we have developed some resources. So we've got some training manuals and training videos that are available on the website. And we've always just a call away, when our volunteers are out if they need help. But most importantly, we want to say a really big thank you to all our volunteers, because every single one of you has made the project possible. And we also want to especially thank and acknowledge our group coordinators. Without their leadership and commitment, we just couldn't run a project on this scale. So, all our volunteers are the heart of the success of this project. So thank you. All right, so on to a bit of information about our sampling. So we're currently in year 1, and we have completed 2 rounds of sampling, but we are still processing the second round of samples. So I won't be talking about the second round at all tonight. The first round involves screening 34 estuary sites across Port Phillip and Westernport, and they're all highlighted by the red dots in this map. For the second round and in the subsequent rounds that will follow, we will be focusing more on around 20 to 25 priority sites each time. So in round 2, we sampled 20 estuarine sites across both Port Phillip and Westernport Bays, plus an additional 3 sites in Port Phillip Bay. And this sampling was carried out in June, July of this year, so fairly recent. As I mentioned, we're still in the process of analysing those samples, so the results for that round will come out at a later date. I do want to note too that we did in this round lose the samplers from Port Phillip Bay. They all kind of sheared off from the buoys. So we have since upgraded the equipment to hold them, and we've redeployed them. So we'll have those sites probably will come out the results in the third round, because they've just gone back out now. On to some results. So in our first round of sampling, we screened for 450 targeted contaminants. So this is the contaminants in our targeted screen. I won't be presenting the untargeted at this point. Of those 450 targeted contaminants, we detected 104 of them. Now, these were grouped into 8 core categories, which you can see shown in the little diagrams on the slide here. So they included things like pesticides, pharmaceuticals, industrial compounds. It is important to point out that these results, as I've just mentioned, reflect that targeted screening method, and this has a strong focus on pesticides. So the list of contaminants in that 450 are really heavily focused on pesticides. So it doesn't mean that pesticides are the only or even the most important contaminants in our waterways. It just means that they're the most likely to appear in from our targeted screen results. As we move into the non-targeted screening, we do expect to uncover a broader mix of chemical types beyond just those pesticides. Also importantly, we don't really find these findings unexpected. The number and types of contaminants we're detecting are similar to results from other studies in the Greater Melbourne area, also nationally and internationally. So when you start screening for large numbers of potential contaminants, you're going to find some, but the value in our data lies in understanding which ones are present and what that means for our estuaries and bays. I will note, so pesticides were by far the most frequently detected. They made up 86% of the contaminants we found. And when you look at the breakdown of pesticides, herbicides are the most common, followed by fungicides and insecticides. Then smaller contributions from those pharmaceuticals and industrial compounds. Now sort of taking a bit of a look at the different chemicals that we found from that screen. Now I know it can be a little bit difficult to read all the names on this slide, but I really just wanted to try and show you all the information about the chemicals that we detected. So these figures highlight the wide variety of contaminants that we did detect from that 450 targeted screen. We found 51 of them appearing in more than 5% of samples. The most frequently detected were fungicides and herbicides, as I mentioned previously, and included 2 fungicides, tebuconazole, carbendazim, herbicides, simazine, diuron, a few other fungicides, propiconazole, azoxystrobin, and again, a few other key herbicides, metlacore, atrazine, propazine. You might have heard of some of these chemicals before. Now, I would like to highlight that, you know, a lot of these chemicals are used in both urban and agricultural settings. And this is a really important point to emphasize that, you know, pesticides aren't only used in agriculture. There's many that are widely used in urban areas. So they can be used around commercial and industrial zones, in consumer products, and also around your home. You know, these urban uses can be significant sources of contaminants entering our estuaries and bays as well. So for instance, if we take a look at the 2 most commonly detected fungicides, so that tebuconazole and the carbendazim, these are widely used in agriculture, and they're used to control things like molds and rusts and mildew in crops, different types of crops. But their use extends beyond farms. So carbendazim is also found in turf and timber products, while tebuconazole is also commonly used in urban environments through home gardens. So you can go buy a product at local hardware, bring that home and use it at home. Similarly, things like the herbicides simazine and diuron, they are applied for weed control not only in places like vineyards or orchards or pastures, which is more agricultural uses, but also along roadsides, railways, and in irrigation ditches, as well as in other urban spaces. As part of another science project that we did a few years ago called the Pesticide Detectives, we did produce some information sheets on some of these contaminants, and these info sheets are available on our website. And we'll also continue to develop new fact sheets on the chemicals we commonly detect. And so these fact sheets just sort of give you an idea of where these sort of chemicals are used, what sort of products, and places you might be able to look to reduce their use, so we reduce their occurrence in the environment. This first round of sampling gives us an initial snapshot of the types and numbers of contaminants that we're finding in our estuaries and bays. Right now, our data really focuses on presence and absence of contaminants rather than concentrations. So this allows us to see which chemicals are occurring at each site and just sort of get a bit of a sense of spatial and then over time temporal differences. And the map that I've got up here is just showing the number of chemicals that we've found at each of our sites. And the green ones are Westernport estuaries and the purple are our Port Phillip Bay estuaries. So every site had detections and we ranged from mixtures of around 6 contaminants at a single site through to 33 contaminants. We found that the Westernport Bay estuaries generally had slightly higher numbers. So they had several sites where we saw more than 28 contaminants at single sites, while Port Phillip Bay sites were a bit lower. So the highest counts were really between 24 and 26 mixtures. Overall, we had 7 sites where there were more than 25 contaminants detected at that single site. And these sites were mostly located near high industrial area catchments or intensive agricultural catchments. So we had a few of them sort of occurring the top of Westernport and then the others the top of Port Phillip Bay. We also had a few sites in Westernport down in this lower section here where we had our really low numbers of detects. And these really reflect an area where we've got lower human influence. It's really important to remember that more detections at a site doesn't automatically mean a worse condition. A site with fewer contaminants could still have one chemical that occurs there at a concerning concentration, whereas sites with many detections might have them all at low levels, but it could also be vice versa. The presence really of the contaminants just gives us a bit of a starting point. It helps us understand where contaminants are more prevalent and can guide further studies to measure concentrations and potential impacts. They also can indicate, catchments where we may need to focus more management to reduce contaminant runoff or stormwater runoff into our estuaries and bays. All right. Before I wrap up and talk about the next steps in the program, I just want to highlight some key messages from this first round of the project. So first and foremost, the contribution of our volunteers is incredibly valuable and deeply appreciated. Your efforts have allowed us to extend our sampling, analyse a larger volume of samples, and also build knowledge and capacity to ensure that our project is going to have impact. It's still early days. This is a 3-year project, and what we're sharing today is just one round out of 9. We're starting to build a picture of contaminant profiles at each site, and we can look then and see, do they change with seasons? Do they change over time? Are there patterns we're seeing, and are these patterns consistent, or are they just one-offs? And so we can start to address all these sort of questions in time as we collect more and more data. The data from our passive samplers provide presence-absence information, not concentrations. So while we've presented results assuming we kind of do say more detections could be worse conditions, as I mentioned just before, even a site with just a few detections could have a contaminant at a high concentration. And that's why in future rounds we will also be assessing toxicity testing. So we'll take waters from the estuaries or bay sites, bring them back to the lab and look at potential impact on flora and fauna. And also in future, we might do some actual concentration measurements. It's really important to remember that this story will evolve as new information, as we get new information. Today we've presented the targeted screening results from that 450 pre-identified contaminants. But our broader non-targeted scans of over 40,000 chemicals is still pending. We plan to apply that broader scan to a selection of our round one samples, and when we pull different contaminants out in that screen that we're commonly seeing, we'll then slowly add them to our targeted list. So that will increase, and when we process our round two samples, we'll have that increased list. The targeted screen was developed over time, as I kind of mentioned before, in collaboration with the National Measurement Institute. It is really fit for purpose, but it's skewed towards those pesticides. So we appear to find a lot more of those. So that's likely just an artifact of what's listed in that screen, rather than an indication that pesticides are the only contaminant present. And I will note that as a rule, as the screen gets larger, there is potential we'll find a larger number of detections at our sites. Despite these caveats, this is really valuable information. It will inform our strategic plans, including things like the Port Phillip Bay and Westernport estuary management plans, also things like the Healthy Waterways strategy and the state of the bays. So it'll provide baseline data and it will help prioritise management actions. And lastly, the program enables informed discussion between all our stakeholders. So that's our community members, special interest groups, traditional owners, councils, policymakers, researchers. And so it can support actionable outcomes. So from things like personal choices that you might make about what you might use at home, to broader management interventions. Our story will continue, and together we'll build the knowledge we need to protect our estuaries and bays. All right, well, where are we off to next? So we've conducted that target screen on round one, which is what I've presented some results to you from today. We're currently working through the non-target screen on those samples as well. And as I mentioned, this could take us a little bit of time because it produces a lot of data. Once that's completed, we will incorporate findings of like new contaminants into our targeted list, and we'll process our round two samples. We will be preparing and sending out a survey to our volunteers to get your feedback on the program and the sampling process. So you'll see something probably come out from Mon about that. We'll continue to build contaminant fact sheets on those contaminants we're finding quite frequently across sites, and they'll be available on our website. We'll continue to provide support to our volunteers through training sessions and in coordinating and running the sampling events. And I suppose really importantly is the third round of sampling for year one will take place in October and November. We'll look at sampling around 20-25 sites, but these are to be determined yet, and we will be getting out as soon as we can some further details to everybody about when that sampling will take place and at what sites soon. So keep a lookout for that, please. Oops, sorry, slip of the mouse. All right, so that's all I've got for tonight. I'd like to thank you all for coming along and contributing to this project. If you'd love to get involved or just like to get involved, you can follow the link on the either the website or through the little box thing. I'm not great with technical things like that. So you can follow those and you can sign up as a volunteer. And that's just, you can sign up to just hear about what's going on with the project, or you can sign up to be actively involved and help us with the sampling out there in the field. And now I'd like to just open up and take questions from any of you.
So we didn't have anyone put any questions in the chat. If anyone would like to know anything further, yeah, you can actually raise your hand and ask it directly of Jackie or the team here. Even comments.
I think we've got...
Nav, if you'd like to unmute yourself.
Hi, everyone. Great presentation, Jackie. I've got a question regarding how have you decided like when to sample, and are you guys also planning to do some PFAS analysis in this project, or is it just going to be those 450 compounds for the targeted screening?
Good question, Steph. So sampling, we do try and sample always at low tide because we need to get the samplers need to stay in the water. So we want to put them as far out into the estuary as possible to ensure that during the tidal cycle they'll stay under water. So sampling is usually during low tide, and then in terms of season, we're just sort of breaking it up to start with to try and cover the different seasons. And it's also been a bit driven by the availability of data to direct the next sampling round as well. That's answer to that part. Part two of your question. So we've got the two screening approaches, and feel free, Palasi or anyone from NMI, to stand in at this point and say anything as well. So the targeted screening is the 450 compound list, and that's our first comparison in each round, but that list will kind of grow as we do the untargeted screening. So the untargeted screening is comparing to broader libraries where there's sort of over 40,000 different chemicals. So this is industrial compounds, fragrances, whole range of different chemicals that we'll be comparing to. And when we start to see some of those frequently come in, we will look at like buying standards and then moving them over to our targeted list if we see them frequently. In terms of PFAS, as far as I know, and this is where I think it would be great, maybe even if Hal or Palasi could stand in, but we was not going to be possibly looking at that because of the way we're sampling, and it's likely we could get some contamination that's not from the waterways. So does Hal or want to comment any more on that? Sorry to throw you in there, Hal. Or Palasi.
Yeah, so with PFAS, because how's available, but I'll just say with PFAS, like Jackie mentioned, some contamination that can happen with the instrument if it's not properly adjusted for PFAS monitoring. At the moment, we are not considering that, but there is opportunity in the coming years if that capability is integrated into the instruments here. Right, Hal?
Yes, so I just want to add that in from what Jackie and Polati just said. With the PFAS one, the chemistry of PFAS is totally different than what we're looking for. So it could be the way that we're collecting the sample at the moment. Maybe it's not suitable to collect all of the PFAS. So that is something that we need to discuss a little bit further with IMIT team to find out whether, and also we need to do some sort of trialing as well to see if the current methodology collecting the sample we're able to see the PFAS. So I think it needs a bit of time to prepare. So for all these contaminations and for the unseen threat, we really have to be really careful of what we look for to. Yeah. Thank you.
Thanks, Howard. Thanks, Pelasti, for jumping in.
That's okay.
These are our analytical chemists. That's not me, guys. I'll ask the experts.
Thanks for that. Thanks for answering it very thoroughly. Look, I've been sampling these 13 rivers out of the 20 rivers that is part of your project and I've sampled in 2022 and 2023. So I've done 3 campaigns, and I've got the data for PFAS, for which I've analyzed for 44 compounds. So it would have been like, if this could happen, it would be a good integration of comparison. So I'm writing that data up, so it will be published hopefully soon. So if you guys are already sampling, it would be good to run that for PFAS as well, so we can build a big picture. Like, so that was my that's sort of my interest, or that's what I've been working on for the last 6-7 years. So yeah, it could potential there.
Okay.
Wow.
I think I have the manager in Sydney lab, Gabby, online. She's the expert in the PFAS testing area for NMI in Sydney. So the work for the PFAS at the moment is being done in our Sydney lab. So I don't know if Gabby be able to have some comments on this testing, and also these comments from you and Imati. Gabby, are you online, or I'm not sure if she's still online.
I am.
Hi, Gabby.
Hello,
I'm
everyone.
sorry to
to
dump
throw
you
you in
in.
the deep end.
That was a great presentation. Thank you very much. So we didn't do any testing on passive filters as yet, but I know that somewhere there are already some passive filters that can be used for PFAS. We were looking for them to see what they are, what are the possibilities to test, because we always have to consider not, the standard list of PFAS. We have to consider actually the big picture to be able to analyze how many PFAS we can. So yes, this is what I can say, not too much, but the technology evolves, so definitely there are passive filters to test out there.
Right. Thanks for that.
Thanks, Gabby.
Thanks.
Yeah, that makes sense. I agree with it. I'll leave my email here, like in case this happens, like you have my contact. So I've been working with Professor Oliver Jones, so
Yeah.
almost finished. So yeah, I mean, be in touch if a collaboration will be really good. Yeah.
Very
Thanks,
good.
Nav.
Thanks, Nav. There was a question put in the chat by Luke, and he's asking, why were pesticides chosen as the initial targeted analysis? So it's a bit of an artifact of our work, isn't it?
It is. It really is an artifact of our broader work that we've been doing over the last 15 plus years. So we've been working with NMI for a very long time on this and developing screens for chemicals of concern in waterways all across Victoria. And as part of that, we have had a large focus on pesticides, but we've got some recent students that were based at NMI that have also been looking at fragrances, and we have looked in some of our programs at other select contaminant groups, and that's where we're trying to use this broader 40,000 chemical libraries to look at things other than pesticides. So those screens will bring in other compounds that aren't so pesticide focused, so they can be pharmaceuticals or some other industrial style compounds that will come up from those screens. So that will sort of flesh out our screen, our targeted screen, a little bit more to be more broad.
Very good.
Thank you.
Well done. There's no other questions in the chat at the moment. Shall we
Did
give
anyone else want to put their
people-
hand up?
Yeah, we're happy to take verbal. Yep, I think we're all good. So it's probably next steps from here, Jackie.
Yeah, so as
Yeah.
I kind of mentioned, the next steps for us are to complete the untargeted screening on those round 1 samples, which Palasi is working with all the team at NMI at the moment to do, and then to process our round 2 samples. And we'd like to get those, some preliminary results from those, and then we'll work out our round 3 sites that we'll target. We will target, I know in round 2 we didn't do all the sites we had in round 1, so any that we missed in round 1 we will pick up in missed in round 2, sorry, we will pick up in round 3, but there'll also be a number of sites that will overlap, and they'll be the ones that we sort of start to get a bit more of a temporal profile on. So by the end of the first year, we'll have at least all sites having 2 rounds of sampling, but some will have 3 at them. And then we'll be heading into round year 2 and undertaking 3 rounds again next year of sampling. Again, targeting different periods and trying to just, improve that baseline knowledge of contaminants. So we'll be letting our volunteers know very soon when that next round 3 will be occurring, because we've sort of started to book in dates that work with the lab and also with the tides. So I've been working on that over the last week, so we'll have some things come out soon on those dates, and as I said, when we know the sites. The other thing is, if anybody wants to sign up, as I said, head to our webpage if you want to get involved. We'll probably hold at least 1 training session before the next round for anyone that missed out in the last round of training sessions. That's our hands-on training on site, where we show you how to handle the passive samplers and how to deploy them and how to retrieve them. So that'll cover sites where we missed last round and didn't sample, so that we can train up volunteers that'll be helping out at those sites, but people will be able to come and join those from anywhere. And the other thing is our report. Oh, I forgot about that part. So we have prepared a summary report on the results from round 1. I was hoping that would be up on the website today, but we've had some minor hold-ups, so I'm hoping it will be out on the website by the end of the week. But we will let you know. We'll send out an email to anyone on the email list when that is available on the website, and we'll also send a social media post to let people know that that's up on the website as well. So keep a lookout for that coming out, and you'll be able to go to the website and download the report and take a look at all the results I've presented tonight.
And probably be able to read some of those smaller names. You can expand them out and read all of the names. Yeah. Very good. The only other thing I was going to mention, Jack, was in this 3rd round of sampling that we're likely to be asking volunteers to get water samples as well for the toxicity test. So that would be at the retrieval stage, but then that will help us look at the toxicity of the water at particular sites, rather than looking at any particular pesticide or contaminant. It's looking at the site water and saying, all right, the combination of what we've got at this site, is that a problem or not?
Yeah. No other questions?
Yeah, or comments. Doesn't have to be a question.
You just remember, if you ever have any queries about the project, feel free to contact us. Our email details are on the website, so you can always touch base with us if you have any questions about the program or the results. Yeah.
Very good. I think we might just about call it a night then.
Thanks, Jackie.
Thank you.
Thank you.
Thank
Great
Thank you all
presentation.
for
Thanks, Monica,
joining.
Jackie.
Yeah.
Cheers.
And I
Thank
think
you all.
it just reinforces the collaborative nature of this project and how important it is with our NMI. Tyler's here from Eco Centre. I think Lisa's here from Estuary Watch, Melbourne Water. Cam Turner, who's been helping us put the
samples in the bay.
Put the samplers in the bay, and lots of volunteers. It's been fantastic.
Right, thanks everyone. It was good feedback from the data. Thanks for that.
Excellent. Thanks,
Great.
Vincent.
Thank you, everyone.